Today we're going to talk about GenAI and how to apply this type of technology in the field of drug discovery, discovery. So I like to compare drug discovery industry to car manufacturing. If you have a look to what has been done in this field 100 years ago, car was made by hand. And right now, if you go to a Tesla factory or other companies, now it's done by robots. But what happened in the field of drug design? Unfortunately, it doesn't evolve too much. This is the lab of Marie Curie, more than 100 years ago. And this is a lab I would say, a classic lab in pharma or industry. So why we have such a difference is just because in the manufacturing car industry, you are producing the same car every day. And in the drug design industry, I would say, in the drug discovery, you are producing different molecule every day. So it's very easy to automate something which is repetition, but it's very hard to automate creativity. In the drug design world, I would say every day you need to think about how making your molecule better, and it depends of the results you obtained before. So that makes the things very difficult. So to identify a new starting material, a new chemical matter for a new targets, usually people tends to use what is called virtual screening. Because the compound are almost on the shelf, at least very easy to make, and they are quite inexpensive. And that's why this solution has been widely used in the field. One of the main drawbacks of this type of approach is that you have absolutely no control on the chemistry. You only get what's available. You have a target, you perform a virtual screening. If in your database of compound, even if it's a huge database, if the solution is not present in this database, you will get nothing. So a few years ago, we asked ourselves, can we find something as efficient as virtual screening, meaning that we can identify rapidly new molecules, but at the same time, is it possible to have the same advantage of virtual screening, meaning that having access to compound which are easy to make and compound on which we can precisely control the chemistry, the starting material. So in 2016, we thought that generative AI will solve that problem. And many papers has been published around that approach, using deep learning technology to generate new molecules. You can work on smiles, you can work on graph, you can work on fingerprints, even selfies or other encoding of the molecules allow you to train a neural network in order to generate a new piece of molecules. This is great, and it has been a lot of excitement around those new technology. And one of the main drawbacks is that this type of technology does not encode for synthetic feasibility. So those type of approaches tends to generate totally unrealistic molecules simply because they don't know what it is feasible molecules. They don't know what it is a crazy connection between two groups of atoms. So things has evolved, and since 2024, we start seeing some paper where they try to embed in this type of algorithm the notion of reaction and the notion of building blocks. But it's still very limited. We have been able to see in all those publications that they are usually, because of GPU memories and difficulties, we still see a database of building blocks which is quite little. We are talking about thousand, ten of thousands of building blocks. We are also seeing a set of reaction which is limited. We are talking about 100,000 of reactions. So it's very limited and it does not cover at all the size of the chemical space. So to summarize, when you want to start a new project, you need to explore this vast chemical space. We're talking about 10 to the power of 60 possible molecules. And there are many solutions to explore that chemical space. You can use a famous traditional method, which are in vitro method like HTS, DER, DNA encoded library, which allow you to explore on the bench a huge chemical space, 10 to the power of 6, 10 to the power of 9, which is already interesting and big. Then, virtual screening, as I said, is now exploring a huge chemical space. We are talking about 10 to the power of 10, 10 to the power of 12. Again, with the difficulty I expressed before, usually the novelty is not very high and you have absolutely no control of the starting point on the chemistry. And you need to screen everything to identify what you're looking for. So it's like kind of a brute force approach. Even though there are heuristics to accelerate that, the concept is still brute force approach. And then, as I said, in 2016, GenAI arrived. Everybody thought it would be a revolution, but those type of approaches, as I said, tends to produce unrealistic molecules. And again, you cannot control the chemistry in either the exit vectors or the building block. So at Iktos, we decided to take another direction and we developed what is called synthesis-aware GenAI. So it's a technology which generates molecules based on building blocks and reaction. So basically, when you generate the final molecules, it encodes the sequence of action, namely reaction and building blocks. Of course, you don't want to generate molecule randomly. You want to generate molecules guided by good fitness function, especially the activity fitness function. We have worked a lot on that in that domain, and I will give some words about that. So the goal of this synthesis-aware GenAI is to design novel, feasible, and active molecules. We have published 2 papers regarding those 2 technologies. We're going to share with you those papers. So I think it's very important to understand that being able to generate meaningful and feasible molecules totally unlock the downstream workflow. If you have a system which generates molecules which are very difficult to make, it becomes difficult downstream then to go fast and to branch any automated tool. So for us, we believe that this generative AI piece is a cornerstone of the whole DMTA cycle. So you need to be very good at that. And at Iktos, as I said, we have developed this synthesis-aware GenAI technology called Machia. After that, of course, you want to see all the possible retrosynthesis routes to make that molecules. And for that, you need a retrosynthesis AI technology like Spire. Once you have identified the routes and you believe that it's a good route, then you need to execute that chemistry on a robot. So you need to take care of translating a list of molecules into a list of robotic instruction. And this is the goal of ILACA. And then, of course, you need to operate that on a robotic platform. Then you need to perform analysis and purification before going to biology in order to close that DMTA cycle. And today we'll cover the 4 first topics: GenAI, retrosynthesis, orchestration, and robotic platform. To be able to do so, you need to break the silo. As I said, if you are generating a molecules which is impossible to make, forget about retrosynthesis, robotics, execution. So first, you need to be very good at GenAI, and you need to generate a listing of molecules which is feasible in the robots. As I said, the technology we have developed is called Growing Optimizer. It's not virtual screening. It's not GenAI text-based. It's a system which has been trained on millions of reactions. It has learned organic chemistry. We have trained the system on the Pistachio database, which is patented reaction, publicly available. And if you train that algorithm, if you train our algorithm on that database, it transforms that into 3.6 thousand name reaction. So it covers everything which has been published so far. And we use a database of 1 million curated building blocks, available building blocks from our preferred providers. If you combine this name reaction plus the 1 million building blocks over several reaction steps inside Machia, you can cover a large chemical space. We are talking about 10 to the power of 27 possible molecules. As I said, it generates feasible and novel molecules, and we can guide this GenAI through different type of fitness function I'm gonna show you right now. In the paper, we published around our technology. We explain how it works. So we have 4 neural network. You have here kind of a RNN, which is a recurrent neural network, which is the backbone of the technology. And then you have 3 other RNN. You have the reaction continuation neural network. You have the reaction type neural network, and you have the building block neural network, meaning that this first neural network decide if we should keep adding a building block or keep reacting on the molecule. So it's a decision criteria to stop or to grow the molecules. So let's say we start from A here. The chemists say, I want to start from this fragment, and I will let the AI taking decision for me. So the chemists say, I want to react on this nitrogen. I want to react on this chloride. That's it. This is the only thing the chemist inputs to the system. Then the AI will say, okay, given that information, I think I need to continue. So this RCNN decide to continue. Given that building blocks, the AI should decide, should I add a building block or should I perform a reaction like oxidation reduction without adding a building blocks? And here it seems that the user expects something to be added here. So this neural network say, yes, you should add another building blocks. And then once the system say you should add a building blocks, the building block neural network decide which type of building blocks. And then you have a prediction which predict a building blocks. And then the reaction occurred. So the building block prediction is this one. The starting material is this one. So we have a prediction, A plus B gives C, and it relays this intermediate. Given this intermediate here, this RNN decide, should I stop or should I go? And this RNN say, okay, you should keep working on that building blocks. And then decide, should I continue? Yes. Which type of building, should I add or not the building blocks? The answer is yes. We have still a remaining exit vectors to react on. And then the building block neural network should decide to add another building blocks. And then the reaction occurred to generate this intermediate. Then this E intermediate arrive here. The RNN decide to continue again. And then now the RNN, given that molecules predict that it should not add another building blocks, but it should perform an A give B reaction. And in that one, it's addition of this acrylamide moiety in this position. And then the compound F arrive here and decide, should I stop or should I go? And then the decision here is to stop, and I get that final molecules. So of course, we are not generating this type of molecules randomly. We use external fitness function. So as I say, those 2 AI generate 1,000 molecules, as I just explained. And then we score those 1,000 molecules again, ligand-based, structure-based, QSAR, models, descriptor, substructure, etc., to have a batch value. And then we send this information back to the system, which is going to change the weight of the AI and generate 1,000 more molecules, either by changing the reaction prediction or changing the building blocks. And we do that iteratively in order to obtain ideal in silico molecules. As I said, we can tackle all the use case because we have trained this AI on organic chemistry, so we know all the reaction possible. So we can grow, we can link, we can cyclize, we can macrocyclize. Everything you may imagine as a medchem, as a conchem, is totally feasible into Machia. So we decided to bench this technology against Reinvent 4, which is the most famous open source generative AI based on smiles. It's not based on reaction and building blocks. And we wanted to bench between this one because it's very well maintained and it has this capacity of doing fragment linking and fragment growing. So it was the first, it's the best open source generative AI. So we wanted to see how Machia compared to Reinvent 4. To do so, we decided to take complex molecules. The one you encounter in your project. We didn't want it to perform a benchmark on very simple molecules because in real life, every drug hunter usually work on complex molecules. So we wanted to have tricyclic pores, aromatic, aliphatic. We wanted to have quaternary centers. We wanted to have microcycles and this type of things. We asked Machia to try to use Tanimoto similarity to find back those molecules. And we asked the same task to Reinvent. And as you can see, we can start from nothing. You let the system try to understand by itself using a Tanimoto distance, try to recover those molecules. And as you can see, it is very difficult for Reinvent to find back those complex molecules. Very often, Reinvent, get lost into potential well, generating molecules with a very low Tanimoto similarity, which is not the case with our technology. We arrive in most of the case to find back the molecules or to be very close to the target without any constraints. So the system start from scratch, has to find by itself the starting material and the reaction. We ask for another task where you impose a fragment, a starting fragment, either the right-hand side or the left-hand side of each molecule. In that case, Reinvent is a bit doing better because you still give some part of the molecule, but still for all the molecules, Reinvent get trapped, contrary to our GenAI, which either outperform Reinvent or in the best case is able to find back the exact molecules. And we perform as well the fragment linking approach. So you give the left-hand side and the right-hand side of the molecules, and you try to see if the GenAI is able to find the heart of the molecule. In that case, we obtain very good results using Machia. We tried as well another task, which is more structure-based task. So we try to optimize 3D scores using this GenAI, either Machia or Reinvent. In 3 different use cases, PIM1, ARC2, and TIAM RD, where we use a starting material, a starting point, like a fragment, and we use template-based docking or anchor fixed docking, and you define those starting material, and you ask for the GenAI to generate something new in this direction, either through Machia, where you can define the chemistry, or through Reinvent, when you just need to put a star on the atom here. And as you can see, for instance, on target 1, ARC2, we generate very meaningful structures with very good scores in 3D, better scores in 3D compared to Reinvent. So this AI is able to find better solution, but not only the score better, but also the quality of the molecules. As you can see here, we get complex microcycle, almost impossible to synthesize. In terms of diversity, also, the pairwise Tanimoto similarity is very low, meaning that comparing every molecules one by one, we have a lower Tanimoto similarity, meaning that we produce more diverse structures compared to reinvent. We obtain Murco-scaffold, high number of Murco-scaffold, meaning that we explore much more in terms of diversity. And if you plot that on a UMAP, you can see reinvent in orange, Iktos in blue. We explore a much broader chemical space. So on all tasks, we outperform reinvent, and I really invite you to read this paper. The code of our approach is available, at least the code of the experiment. Of course, the code of Micah is proprietary. On top of that, I think it's very important to fulfill as well what chemists expect from such solution. Chemists want to use it and to define precisely what they want to see. So for instance, in Micah, you can define your starting points. You can define the exit vector. Here, you want to start with a bromo. You want to start with a nitrogen. It's not like a star and you let the system grow. You really decide, I want to start from bromo. I want to start from nitrogen. And then you can define as well the chemistry you want to involve in this position, the chemistry you want to involve in this one, and just let Micah generating against different type of scores. You can do the same with the scaffold hopping. If you're looking for a new central score, you keep the left-hand side, the right-hand side, and you let the system finding the good scaffold and the good sequence of reaction to find that new compound. You can also perform more complex tasks where you are targeting heterocyclic chemistry, cyclization. You want to start from here, playing with alpha bromoketon to generate imidazole thiadiazole. It's very easy into Micah. You define your exit vectors, you define the constraints on the building block, and the system will generate totally new molecules. We can tackle also very complex use case. We can play on macrocyclization if you're working in that field, and of course, still using 3D reward. We can use Micah in the field of PROTAC. For instance, here it's a sequence of our AI. The chemists say he wanted to keep the left-hand side, he wanted to keep the right-hand side, and Micah automatically detects building blocks, make chemistry on building blocks, make it react until to generate virtually this final PROTAC. The output of Micah, it's a listing of molecules, it's a tree of synthetic forward synthesis, it's a 3D scores. You can also play with name reaction. And this is very important because today we are talking about GenAI done right, meaning that you want to generate something which is feasible. So you have also control not only on the exit vector, but also on the chemistry you want to use into that generating process, but also the chemistry you want to avoid into that generating process. Generating molecules, it's a good thing, but generating the good molecules, it's even better. So we have developed a flow-based generative alignment method. It's our Iktos post-prediction approach. As long as you have a reference molecules, Iktos technology is the fastest and the best approach to generate molecule in 3D. This core technology has been published in NeurIPS last year. It allows you to generate molecule using 3D LB or 3D SB approaches. In 3D LB, we have showed that it outperformed by far LS-Align or Rochambeau, even for very complex tasks. I think this technology is very well suited for flexible molecules, PROTAC, macrocycle. It's very efficient to deal with those type of complex structures. As you can see, we have a very low RMSD compared to existing published ligand-based approach. In the field, you can also customize your pharmacophore, so you can put weight on some position of those molecules. As you can see here in macrocycle, in cyclic peptide as well, you can define hydrogen bond donor, lipophilic area, and the system will generate new molecules with very nice overlap. As you can see here, we even do not detect the reference molecule since the alignment is really good. It's the same here in product. You see on the left-hand side here, we have very nice alignment. On the right-hand side, very nice alignment. And here I have a new linker mimicking the shape. So it's the best technology to align molecules together. If you have a pocket, if you have a reference, we call that precise AI docking. We obtain the best performance compared to existing technology on publicly available. If you don't have a reference, you have only a pocket. We call that AI docking, and we perform as well very nice results. I would say that we have similar performance compared to AlphaFold 3 or Interformer, for instance, but we have a better RMSD and we are very, very fast. It takes less than 2 seconds to dock using this AI docking technology molecules. So let's say that now we have a very nice listing of molecules. We now need to have a good roots and good retrosynthesis roots. So for that, we use Spire, which is based on the same database of Machia, 6 minutes reaction. It gives you 3.6 name reaction, the same number. But inside Spire, we have more than 256 million building blocks coming from 44 preferred providers we work with. So Spire, it's a data-driven approach. You give a database of reaction. In our case, it's Pistachio. You give a database of starting material. And the system is trained in a couple of days. We train different type of AI. You have the disconnection prediction. You have the search strategy. You have a reaction feasibility. All those AI algorithms make sure that it cuts the molecule properly. It makes sure that the two daughter molecules will react properly, avoiding chemoselectivity, radioselectivity issue. So you have automated strategy of protection, deprotection, and it gives you a tree. Spire is publicly available. It's free during 2 weeks. You go to spire.ai, you create an account, and you can test the technology. Spire is quite unique in a sense that you can really customize the retrosynthesis search, either by playing on the price of the building blocks. You can also play on the delivery time, the providers you want to use during that retrosynthesis search. Like Machia, you can play on the name reaction you want to use, reaction you want to avoid. You can play as well on key intermediates. You have a list of starting material you want to use into that research. It's very easy to do so. Also, in terms of presence, absence of substructures, you can also use it. You can also define the bonds you want to break during that retrosynthesis or the bonds you want to protect. Spire is available through Spire frontend, GUI, or through API if you want to use Spire and this retrosynthesis into your in-house workflow. Once you have good molecules, good roots, you need to select the best one. So for that, we have built Ilaka. Ilaka take as input molecule 1, 2, 3. It can come from Machia, it can come from your own design, your chemist, your virtual screening. Ilaka is totally agnostic of the molecules you are giving to it. And then Ilaka will trigger Spire API. For each molecules, we're going to find plenty of roots. All those roots are compatible with your robot because, as I said, you can define your preferred providers, number of steps, and the chemistry. And then the question is, which route should I select to run my chemistry? And this is where our scheduling AI makes a decision, take the route number 4 of the molecule 1, the route number 1 of the molecule 2, the route number 2 of the molecule 3, etc. The orchestration algorithms select those routes because they share similar temperatures and similar duration. So you can batch those reactions together on the robot. Really, the goal of our robotic platform is not to run 100 Suzuki coupling, but it's to run 100 reactions, totally different from each other, but they share a similar duration and temperature. Then we show that to the chemist at Iktos, and then the chemist can approve or reject this organization, can define the quantity he wants, etc., etc. Everything is like an ELN pre-filled automatically. It takes less than 1 minute to validate that. Once it's validated, we can place ask for a quote to our preferred providers, and then the provider receives this type of interface, give us the price and the delivery time. We can sit it into our platform and accept this quote, and then we receive those starting material pre-weighted. It goes to our Hamilton, which is like a big fridge where you can put all your starting material, and then it updates automatically our inventory. Then the chemist can tell to Ilaka, okay, today, what can I do given all the molecules I want to make and given all the starting material I have received into my Hamilton, what should I do today? And this is what Ilaka tells. It tells, okay, this is all the starting material you need to put on the robot. This is all the catalysts you need to put on the porter rack. And this is also all the solvents you need to put on the platform. And then the chemist can place those starting material on the platform. You can place it randomly. The system will pick each of those starting material and then will identify the position and the barcode. So now we have, again, good molecules, good routes. We have been able to select the good one, to place the order of the starting material. Everything is set. I just need to launch my robot. So it's a ChemSpeed robot where we have two reactors. So 96 vial in total of 8 milliliters. So we have two reactors so that we can run two types of condition in terms of temperatures, which is good for batching our campaign. Here, this is where you put the starting material. So if you have A plus B give C as a reaction, you place A and B here, and then you have the solvent. The solvent are added to the reactors, and then the mixtures is adding to here the two islings, the two reactor on the left and the right hand side. Then it's shake it. You can also add catalyst here with the GGU. And once the reaction is done, we perform a workup on a supported phase extraction. This robot can run 96 reactions in parallel. One chemist can operate one robot. And we can run up to 500 reactions each week. So it's very productive, and it's also scalable. If you want to put more robots, Ilaka can deal with plenty of robots. So we use this platform on real-world use cases. We have published recently in Chemical Science our approach. We call that cluster synthesis. So we explain this original approach to run campaign of reaction for reaction which can be batched given the reaction direction. So this approach allows you to run Suzuki coupling, Mitsunobu coupling, Amide coupling, whatever reaction on the same platform the same day. So it unleashes the capabilities of the platform. So for that, we use in-house inventory, in-house chemistry, and we ask Maka to generate a library. So in that case, it was a library of 135 molecules. And we show in this paper how we have been able to make those 135 molecules in few days with minimum human intervention, everything organized automatically. I really invite you to read that paper. Today, I will present unpublished results where we show how to use that in a real drug discovery project. In that case, it was PKMYT1 case studies. It's a kinase from the WEE family. And the idea was to, okay, can we identify a new scaffold compared to the competition? If you have a look to PKMYT1 targets, one of the lead series has been patented by Repair Therapeutics. It is in phase 1 right now. It's non-receptive. As you can see, these very nice molecules. If you see other repair patents, you have other type of scaffold. But if you look closely, you see the same type of groups where on the right-hand side, you have a phenol, as you can see here. On the left-hand side, you have hinge binding motif. In that case, it's an amide moiety. We have it here as well, and here engage in a ring. And also you have this NH2 moiety in this position. Then the scaffold here is different. Of course, the goal is to have freedom to operate. And it is how we can see Generative inside our 3D technology inside Machine. So we ask ourselves, okay, who are the players worked in that field? And if you see, there are a lot of followers on the Generative, Dan Atlas, Simred, Insilico, Engsu, Akriven, etc., etc. As you can see, all those guys use the same approach, a phenol on the right-hand side, an amide either engage in a ring or not, and they try to pattern bust the scaffold, as you can see here in the middle. What you can observe is that they all contain this NH2 moiety. And why they are doing so is because if you remove this NH2, Repair showing the patent that you go from 22 nanomolar active compounds to a totally inactive molecules. So it seems that this NH2 is mandatory for activity. So we say, okay, let's take this as a challenge. Can we find something without this NH2? And let's keep the phenol, let's keep the hinge binding motif, but can we find something without this NH2? And we ask Makia to do so. So on the left-hand side, we keep the phenol, and we ask to generate something new in the right-hand side, starting from the bromo derivatives or the boronic acid. Then inside Makia, you can, as I said, define what you're looking for. Here we wanted to make the hinge binding interaction. We wanted to do a pipe stacking here with the scaffold, and we wanted as well to place this phenol moiety into this sub-pocket here. And then you just press go, and the GenAI has been able to generate 13 new scaffolds. 16 molecules have been synthesized in our robot because they were very easy to make. And we have been able to identify 4 active scaffolds. So as you can see, we get the phenol on the right-hand side because this is something we ask for to Makia. And then Makia identify, 4 new scaffolds, not published yet, without the NH2, having very nice activity. As you can see, 003 has a 30 nanomolar activity, and the others are sub-micromolar activity. And it was in only 1 DMTA cycle. So it was very efficient approach. At the same time, in terms of diversity and novelty, it's a success, but also in terms of synthetic feasibility and synthetic access, only 2 reaction steps done on our robotic platform. Then, of course, we wanted to explore the left-hand side and the right-hand side. We knew that the phenol was the best moiety, given the publication, but still we wanted to explore that position. We didn't improve the first hit. On the left-hand side as well, we wanted to see how it behaves. No improvements, even though we still keep nice active molecules. And then we explore the south part here and the pyrazole moiety. And we have been able, again, using Makia and using our GenAI, to identify a new interesting group, making an interaction deep inside the pocket, increasing the activity to 5 nanomolar activity. So as you can see here, using a good methodology to generate molecules, a good 3D scores, and then downstream workflow, a robot, an Ilaka, which is able to rapidly place orders and to launch the chemistry on our platform. In less than 3 months, we have been able to do the structure-based work, the GenAI, and the synthesis, and the testing, and to identify those very active molecules with a totally fully novel chemotype compared to the whole competition, including Repair, but also all the competitors. This technology is available either through softwares with Makia and Spire. We already have several customers using that tech. The retrosynthesis technology is either available through Spire. As I say, it's publicly available on spire.ai. You can create an account and test the technology. The engine as well is available into Reaxys. We have trained our AI on the full database of Reaxys available through their platform and through their interface, but powered by our AI. We are also engaged in a very nice drug discovery partnership where our partners come with the targets and then we use our tech ourselves to help them identify rapidly chemical matter in their project on very challenging targets. And of course, we also use our technology for our own needs. We like to work on challenging targets like MTHFR2 involving inflammation and autoimmune disease. We are also working on amylin receptors, obesity, metabolism. We start this project recently. We already have hits on amylin receptors, and we also work on PPI interaction to leverage our proprietary platform. We have a biologic platform where we can assess PPI interaction in cellulo. It's a very original and nice approach. We are right now in the hit discovery phase of that project. If you're building your own labs, your own LLM, your own agentic lab, our technology is also available through API. So you could, if you want, still use that technology without the front end, but directly inside your own platform, in your own workflow, because everything is available through API. This is a new offer we are proposing. If you're interested to test our API, just come to us and we will make the API accessible to your platform. You will be able to design, to score, to decide, to synthesize directly inside your pipeline. Soon we will do a webinar with ChemSpeed because we will announce today a partnership with ChemSpeed where we bring our AI and ELACA technology on top of their hardware and softwares. If you're interested to build such a platform, just come to us, come to ChemSpeed, and we can start discussing your setup and what do you want to do and see how we can help you in that regards. I want to thank you all. I want to thank you, Iktos team. We are more than 55 people, scientists across the world, in France, in US, in Japan. We have today Victoire and Mizuki, which can take your question in Japanese, which are based in Japan. Feel free to reach to them. We are a team of scientists in the field of chemistry, medicinal, computational chemistry, in the field of biology, drug discovery, machine learning, AI, HPC, cloud, softwares. We assemble all those talented people around that goal, which is to discover new molecules for the benefit of patients. This is supported by our VC, M Ventures, Debiopharm, Omnes, but also thanks to France, with BPI France, Europe with the EIC, and we are also part of the French Tech 2030. And I want to thank as well European Union who finance the robotic platform. We have been awarded from the EIC, European Innovation Council, which is a very challenging ground, and we are very happy to have received this grant to improve and accelerate the robotic chemistry platform. Thank you very much and happy to take questions.